Good morning, everyone. Yes, my name is Claire GUISSART. I'm in charge of the routine molecular diagnosis of ALS in Nîmes Hospital. And so I started this project now 1 year ago because of a frustration. I wanted to have a tool that helped me better classify splicing variants, and that's why we developed this approach. So why is there a need to study intronic variants? Well, it's because they represent 11% of variants that are identified in routine diagnostics, and most of them remain in the class 3 categories following the classification of ACMG, which is the variants of uncertain significance. So we needed a tool to help us clarify their functional impact, and that's why we developed targeted RNA-Seq. So it's a very simple project. So here you can see the overview with all the people that are involved in the different steps. And so we have 3 main objectives. The first one is to develop bioinformatic pipelines for routine ALS genetics diagnosis. And we want thanks to that, to improve the classification of splicing variants and solve as many cases as possible. And finally, we want to identify new mutations and study their functional impact. So basically what we ask is another blood sample taken in a packaging tube, which will preserve the RNA during the transport from the prescription center to our lab in Nîmes. And then we perform DNA-seq sequencing. It's part of the routine of the diagnostics. And we compare with the results of RNA-seq. So just a quick reminder of how we interpret splicing variants in diagnostics. So it's a slide with the basic principles. So first, we detect a variant at the DNA level. And so, for example, here you have a mutation that affects a splice acceptor canonical site. And whenever you see a minus or plus, so when you see minus, it means that we describe an intron variant that is located before the exon. And when you see plus, it's after. All right, and the crucial positions are the -1 and -2, or +1 and +2. And we know that if we mutate this nucleotide, it will be deleterious. So then we perform in silico prediction with splicing algorithm to help us to see if there is a destruction of the splice site or a creation of a new one. And then we apply classification according to ACMG guidelines. So when canonical splice sites are involved, it's easier to classify. But when you have a mutation that is deep down an intron, it's often uncertain and it requires RNA evidence. So then we perform RNA-seq, and we can see a different kind of impact on the transcript. Sometimes it can be exon skipping or the alternative use of a splice site, different splice sites. You can also have intron retention or the creation of a pseudoexon. And we have to ask then what impact is expected on the protein at the protein level. Will it be a loss of function or the expression of a mutant protein with the slightly different sequence or truncating protein? And also we have to be very aware of the gene that is involved. Is it a gene where loss of function is a known disease mechanism, or is it a gene where it's more toxic gain of function? So to date, we have included more than 400 patients with ALS, and we performed 80 RNA-seq. And among these 80 RNA-seq, we included 37 patients with splicing variants that were detected at the DNA level and that we wanted to study. And thanks to RNA-seq, we were able to confirm that 11 of these 37 variants were actually deleterious. One of the big challenge of this project is the optimization of a bioinformatic pipeline. So this work is performed by Corentin Marco, who developed a tool which is called splice variant RNA. And he's helped by Dr. Anthony Bourreux in Montpellier, and also the community of bioinformaticians from the different French hospital, BioInfoDiag, which is very active community. And so we developed this tool that combines statistical splice junctions and non-statistical splice junctions and generate not automatically a report with a sashimi plot and HGVS annotation. And we train the pipeline with the biologist feedback. So whenever I detect a splicing anomaly that is not present in the report of Corentin, we adjust the filter and we create a feedback loop to optimize, and then we reanalyze all the patients. So now I'm going to give you a few examples, case studies. So for example, this sporadic case followed by Dr. Fortanier in Marseille, a patient, 51-year-old man with distal asymmetric quadriparesis. And we identified a mutation that affected the canonical acceptor site of the exon 10 of HNRNPA1 gene. And here on this slide, you can see the result of the in silico prediction. So the green boxes here disappear with a C. So this means that the variant is predicted to completely destroy the acceptor site. And so this is the result of RNA-Seq. So it's called sashimi plot. The patient is in red and the control is compared to the control in blue. And as you can see, a simple variant are sometimes not so simple in the impact. And you can see three kinds of effects. The first one is the exon 10 skipping. But you have also the use of a cryptic acceptor site into the exon 10. And also, we detect an intron 9 retention. And these three impacts actually leads to a frame shift and the apparition of a premature termination codon. Another example, so in 2022, we identified a deep intronic variant in SQSTM1 in a patient followed by Dr. Debré and Dr. Bernard in Lyon. And at that time, the ROSA project was not started, and we were stocked in the class 3 category. The variant was of uncertain significance. And this year, we identified the exact same mutation in an unrelated sporadic case, a 77-year-old woman. And she was followed by Dr. Nathalie Guy in Clermont-Ferrand. And she was included in the ROSA project, so we could investigate this variant. So here is the prediction, the in silico prediction. As you can see, the variant is predicted to create a cryptic donor site. You see here the blue boxes. And so we confirm that this cryptic donor site that was created because of the variant was actually used. So here on the sashimi plot, you can see that it creates the retention of the first 109 nucleotides of intron 6, and this leads to a frame shift with the apparition of a premature termination codon. So we reclassified this variant as likely pathogenic, and it helped us to resolve also the case 1. So future developments are mainly the improvement of bioinformatic pipelines, because for now there's some splicing events that are not detected. So, for example, intron retention, it's not detected by the pipeline. And also because it's targeted RNA-Seq, quantitative defects are not well detected. So we want to try to improve this pipeline and to enhance blind detection of splicing defects, because for a lot of patients, there's no variant that are identified at the DNA level. So we would like to see if we can detect splicing anomalies blindly thanks to this method. And so finally, as you can see with the few examples I gave to you, it's really important to test sporadic ALS, because historically they were less deeply investigated than familial cases, but they continue to yield critical genetic insights. And also it's important to integrate systematically the RNA preserved material into the routine diagnostics, because we can retroactively resolve cases. So, and finally, I think that genetic diagnosis is the first step towards the treatment discovery, and so it's important to combine these findings with fundamental research and go for much further in the treatment discovery. Thank you very much. I would like to thank the patients and families, the clinical teams involved, the laboratory staff in Nîmes, the INM team for the scientific discussions, and FieldSlan also, and last but not least, the funding partners, ARSLA and Nîmes. Thank you very much.
Thank you very much, Claire. So we have time for a couple of questions.
Thank you for your presentation. You mentioned that intronic mutation may have a deleterious effect on splicing, but there are also exonic variant,
yes,
which may have a deleterious effect on splicing, especially close to the intron-exon junction, including synonymous variants. So did you look at this also?
Yes, we always check the in silico prediction, even if it's missense or
synonymous.
Yes, and we have some example with splicing defects due to missense mutations.